Hi everyone, today we're going to look into proteins, which is a part of the IB biology syllabus. Now, as you may recall from IGCSE, proteins are made of amino acids. More specifically, they are amino acids that are bonded by peptide bonds, thus form polypeptide chains. Now, the amino acid structure is as follows. We have the amine group, the variable group, also known as the R group or the side chain. We have the hydrogen, and we also have the carboxyl group. Now, although we generally denote the amino acid structure as this, when amino acids exist in cells of the body, which generally have a pH of around 7 to 7.4, they don't necessarily exist in this format. They may exist in a zwitterion format, which would have the amine group to be the ammonium group, the carboxyl group to be the carboxylate group, and they would have charges. But again, that's a conversation for later discussion. Now, when we combine two amino acids together, we form what is a dipeptide, and we also produce water. And that is because we undergo this reaction that's a condensation reaction, which forms a peptide bond between the two amino acids. And what I mean by that is, in the carboxyl group, we will remove the OH group of it, and then from the amine group of another amino acid, we'll remove the H of it. And therefore, we will end up with a byproduct of water, also known as H2O, and a peptide bond right here. And the type of reaction of this is a condensation reaction, which is essentially forming polymers from monomers, in this case amino acids, forming proteins, and via the removal of water, and water is a byproduct. Oh, by the way, the opposite of condensation is hydrolysis, and they are kind of cases of reactions under a large type of reaction called catabolic reactions and anabolic reactions. Catabolic reactions having to break down polymers from monomers to monomers, and anabolic reactions building up polymers from monomers. Right, okay, and so we call a single amino acid a monopeptide, two amino acids dipeptide, and then 3 amino acids tripeptide, tetrapeptide, so on and so forth. And so we call multiple amino acids bonded together a polypeptide chain. And we usually use polypeptide interchangeably with proteins, but we generally use the term proteins for polypeptide chains that have 50 or over amino acids. So, right, how many amino acids are there? Well, the textbook definition and what we learn in IB defines the number of amino acids to be 20. However, something that we need to know is that there are around 500 amino acids in nature. I think there are 22 that can be genetically encoded, but then we generally narrow this down to 20 amino acids. And so, in terms of amino acids that can actually be synthesized and expressed as proteins, let's say that we have 20 amino acids. And these 20 amino acids divide into essential and non-essential amino acids. And what determines whether a single amino acid is essential or non-essential is essentially whether we can synthesize the amino acid. So, an amino acid that we can synthesize would be called a non-essential amino acid. One that we cannot synthesize would be called an essential amino acid, which needs to be intaken by diet. For plants, they can all synthesize all of the amino acids. However, for animals, I think we have 11 non-essential amino acids and 9 essential amino acids that we cannot synthesize, thus must be taken in by diet. Examples of non-essential amino acids that can be synthesized are alanine and tyrosine. Alanine is, I think, synthesized by pyruvate. It's made up of pyruvate. And tyrosine, I think it was phenylalanine, phenylalanine, and then it combines with the hydroxyl group in order to form tyrosine. So these are examples of non-essential amino acids because we can synthesize them in our body. Oh, right. We also have the hierarchy of protein structures. And what I mean by that is we have, because for proteins, they can divide into primary, secondary, tertiary, and quaternary structures, depending on the number of polypeptide chains that they have and the types of bonds that bind these polypeptide chains together. We classify them into 4 different hierarchy levels. Now, in primary, we only have one polypeptide chain. So all of these individual amino acids linked by peptide bonds, and we only have one chain. And we usually say that the end of the polypeptide chain that has the free ammonium group, that entire amino acid is called the N-terminus. The amino acid at the other end, which has the free carboxylate group, it's called the C-terminus. And as you can see, it's one single polypeptide. Now let's move on to secondary structures. For secondary structures, we have alpha helixes and beta pleated sheets. Oh, and by the way, the only type of bonding that's used in primary is peptide bonding. For secondary structures, we have alpha helixes and beta pleated sheets. And the kind of differentiating factor between primary and secondary structures is that for secondary structures, we have hydrogen bondings that cause these proteins to kind of come into 3 conformations. And for alpha helixes, they essentially have hydrogen bondings between the carboxyl group and the amine group. I think it was every 4th, but it's essentially between hydrogen bonds forming between the carboxyl group of one amino acid and the other non-adjacent amino acids amine group. And although when we write it out, we usually say that it's the carbonyl group that bonds hydrogen bonds with the amine group, but it's a part of a carboxyl group. And that's alpha helixes. Same goes for beta pleated sheets. They're slightly different in the way that they're coiled, but same in that they use hydrogen bonds. Next, we have tertiary structures, and tertiary structures differ from secondary structures in the sense that although they only have one polypeptide chain, the types of bondings that are involved are slightly different. And also in secondary chains, secondary protein structures, we only have hydrogen bonds between the amine group and the carboxyl group, but in the case of tertiary, what usually involves in bonding, the type of groups that involve in bonding are usually R group. So it's mostly R group interactions that kind of account for most of the bonding. And so we have R group interactions, which form hydrogen bonding, hydrophobic interactions, disulfide bridges between cysteine groups, and ionic bonds between charged R groups, oppositely charged R groups. And so we have different types of bonding that are involved in tertiary structure polypeptide. Now, a quaternary structure. Now, a quaternary structure is made of several subunits, which are all individual polypeptide chains. So you can kind of think it as having multiple blocks of these tertiary structures all together. And by the way, for your reference, this is a hemoglobin, and so we have 4 subunits kind of together. And also, we can call this a conjugated protein because it has a prosthetic or a non-protein group inside of it, which is the heme group in this instance. So for quaternary structures, we can differentiate quaternary structures from the rest of the other 3 structures in that they usually have more than one polypeptide, or either they're just conjugated and they have a prosthetic or non-protein group inside of them. And alright, I think that's pretty much it for proteins. In our next lecture, we'll be looking into denaturation and also more of the specific properties of all of the R groups.